Dear ladies and gentlemen, welcome to online seminar 4, the CO2EXIDE Electrochemical Flow Cell. It's a big pleasure to present you our activities. Let's start with the outline. At first, the generic CO2EXIDE cell concept will be presented. In a second step, the small size cell with 25 square centimeter active area will be shown. This cell was thought to gain first experiences with the construction principles and with the material selection. In the third step, the large size cell with 300 square centimeter active area will be explained. This cell shall be a part of the demonstrator unit. The CO2EXIDE electrolyzer cell is developed for the anodic conversion of water to hydrogen peroxide and the cathodic conversion of CO2 to ethylene. The generic cell concept of the CO2EXIDE cell is based on a three-chamber design. On this slide, a picture from Dean et al. Is shown to illustrate the three-chamber design. Starting from left to right, it's clearly visible that there is one chamber for the CO2 gas, one chamber for the catholyte, and one chamber for the anolyte. The CO2 gas chamber is separated by a gas diffusion electrode from the catholyte. The catholyte and the anolyte are separated by a membrane. The cathodic copper catalysts that were developed within the CO2EXIDE project were applied to the porous GDE, and the anodic catalysts were directly brought onto the anodic electrode. As mentioned before, the development strategy of the CO2EXIDE project foresees one small cell with 25 square centimeter active area, and in a second step, one large size cell with an active area of 300 square centimeter. For illustration purposes of the generic cell concept, an explosion drawing is shown in this slide. It can be seen that the generic cell concept is based on a number of different individual parts. The individual parts of the cell will be explained in the next slides. At this point, it shall be mentioned that the anolyte and the catholyte flow in the same direction. However, both electrolytes cross each other within the cell. The CO2 gas flow is directed in opposite direction to the electrolytes. CFD simulations were used to optimize the pressure drop and the flow homogeneity of the electrolyzer cell. That means a low pressure drop and a highly homogeneous flow distribution were aimed for. The velocity distribution of the anolyte flow field and the CO2 gas flow field are shown in this slide. At this point, the CFD simulation results of the 25 square centimeter cell will not be discussed in detail due to the fact that later on the results of the CFD simulations of the large size cell will be explained in detail. Furthermore, there are two pictures on the right-hand side of the slide illustrating the view in the real hardware of the 25 square centimeter cell. After the production of the hardware, the 25 square centimeter cell was assembled at Siemens and intensively tested. More than 10 different anode and cathode catalyst configurations were tested, showing the generic suitability of the construction and the material selection. No internal leakage was observed during the complete testing procedure. Therefore, the 25 square centimeter design was evaluated to be a suitable starting point for the development of a large size cell with an active area of 300 square centimeter. This slide shows an explosion drawing of the cell with an active area of 300 square centimeter. The cell was assembled as is shown here. Let's go a little bit into detail. The base plate number 1 as well as the clamping plate number 14 were made of aluminum. POM was used for the end plates number 2 and number 13, the anolyte plate number 4, the catholyte plate number 7, the GDE plate number 8, and the CO2 plate number 10. The anode number 3 can be equipped with iridium oxide coated titanium substrates or with boron doped diamond coated niobium substrates. The flow grid number 5 in the anolyte and the catholyte chamber was made of PEEK. Nafion 117 was used as membrane number 6. As GDE number 9, the Freudenberg GDE type H23C2 coated with copper was chosen. The GGE was contacted by a copper element number 11. A window number 12 made of polypropylene was chosen to make the reaction in the CO2 gas chamber visible. Also, the electrolysis cell with an active area of 300 square centimeter was optimized by CFD simulations. On the left-hand side of the slide, the CFD simulations illustrating the pressure drop in the catholyte chamber is shown. The catholyte is entering the chamber on the lower left side and is leaving the chamber on the upper right side. It's clearly visible that the highest pressure drop is located at the inlet and the outlet of the chamber. The overall pressure drop is around 300 Pascal. The right-hand side of this slide shows the flow distribution over the chamber in dependence on the port size diameter. From this graph it can be seen that with increasing port size diameter, the mass flow distribution is improved. With a port size diameter of 30 mm, a deviation from the mass flow distribution smaller than 10% was observed over the width of all channels. Based on this result, a port diameter of 30 mm was chosen for all chambers. That means for the anolyte, the catholyte, as well as for the CO2 gas chamber. Besides the optimization of the port size diameter, also the complete flow field of the chambers was optimized. This slide shows the results of the velocity field and the anolyte respectively catholyte mass flow distribution. The flow field was optimized to reach a highly homogeneous flow distribution within the active area of the cell. It's clearly visible that the maximum flow deviation is smaller than +8% and the minimum deviation is larger than -4%, which assures a highly homogeneous mass flow distribution. Furthermore, vortex structures can be seen behind the supporting structures at the inlet port. The CFD simulations resulting in the velocity field as well as in the mass flow distribution of the CO2 chamber is shown in this slide. As in the case of the catholyte chamber, also in the gas chamber vortex structures can be seen behind the supporting structures at the inlet port. The highest pressure drop is located at the ports due to the size of the flowing diameter. The highest maximum flow deviation is around 10%, the lowest flow deviation is larger than -5%. That means a highly homogeneous flow distribution is also expected in the CO2 gas chamber. The cell setup for the demonstrator, including the periphery components, is shown in the slide. In the centrum of the assembly, the 300 square centimeter cell is located. Below the cell, the electrolyte supply, inclusive the pumps, is installed. The electrolyte supply is equipped with a filter unit. On the right side of the electrolyzer cell, the power supply for the periphery devices and the theometer is located. On the left side of the electrolyzer cell, the CO2 humidifier and the electronics box is installed. The fume head is furthermore equipped with an optional CO2 humidifier refill and a product gas and cooling and drying spot. This slide shows the periphery of the 300 square centimeter electrolyzer cell of the real hardware in detail. In the centrum of the assembly, the cell is located. C number 1. The electrolyte supply number 2A and C is installed below the cell. The product gas, cooling and drying is located at number 3. The box for the electronics is installed at number 4. The position 5A and C are foreseen for the electrolyte pumps. Thank you very much for your kind attention. I hope you enjoyed my presentation. Please contact Dr. Kerstin Wiesner-Fleischner and me for further technical questions, and for all other questions, Dr. Anna Roth from Fraunhofer.